we did first we did denture structure, then we did anatomy of digestion, like the parts, intestine, large intestine, small intestine, stomach, and all that, and even anatomy of gut. Then we did the okay, no issue, Sam. Then we did with like digestive glands, and we did till the liver. And if I am not wrong, we have to start from the pancreas onward. Yes, am I correct?
Yes.
Yes. So fine. So let's start from the pancreas. This is the last digestive gland. After the pancreas, we will start with the digestion process. Like how does digestion takes place? Means digestion of carbohydrate, digestion of fat, digestion of protein, digestion of nucleic acid. These are the four components which we are going to cover over here. Then we will do the process of absorption, then the disorder of digestive system. And if we will cover all these topics within 2 hours, then we will start with the next chapter, that is the respiration, right? So let's start with the pancreas. Okay, so before starting, I would like to show you a diagram of pancreas. So pancreas is a gland which have both function, like it is endocrine as well as exocrine gland. Endocrine glands are those which do not have duct, means they do not have tube, and they just secrete hormones. Exocrine glands are those which secrete which secrete hormones, right? But this is the gland which do the both of the function, enzyme secretion as well as hormone secretion. Means this is exocrine as well as endocrine gland, right? So these kind of the gland which perform both of the function, these glands are called heterocrine glands, right? So pancreas is a type of heterocrine gland. So this is the diagram of pancreas. Look the diagram, then I'll explain more point for you. Yes. Look, this is the pancreas. And this is stomach, and this is bile duct. It comes bile duct, and as we have discussed, this is the hepatopancreatic ampulla. This part is pancreas. Pancreas have two type of this. The endocrine part of pancreas. These star kind of the cells, they are called isolate of Langerhans. Actually, they are responsible for secretion of insulin and glucagon, right? So we'll discuss all these hormonal part in endocrine part, that is chemical coordination, and that is the actually last topic of our digestive system. And like next thing, that look, this is the actually these are the this part, these glands. There are two kind of the gland. Duct cells, they secrete aqueous NaHCO3 solution, and mainly the main part is that acinar cells, means there are the cells of acini. They are the digestive, they secrete digestive enzyme, right? So cells of acini, they have exocrine part and they secrete digestive enzymes, and this digestive enzyme is poured inside our duodenum part, right, with the help of this pancreatic duct, right? So that's how they function. And what are those enzymes which are secreted by these cells? So let's start. So there are certain enzymes which are secreted by these cells of acini, right? So acinar cells means cells of acini. So let me write it down. Cells of acini. So cells of acini are actually enzyme secreting part, means they have the exocrine function, and the enzyme they secrete. So there are certain proenzymes, means inactive enzyme. So there are 3 proenzymes. So 3 proenzyme. So what are those proenzymes which are secreted by cells of acini? So first one is trypsinogen, right? Second one is chymotrypsinogen. Chymotrypsinogen. Both are responsible for digestion of protein. Then there is one more enzyme, that is procarboxypeptidases. Procarboxypeptidases. Look, these are proenzymes. These all three enzymes, they are inactive kind of enzyme. They are inactive enzyme. Trypsin will become active and it will be converted into trypsin. Trypsinogen will convert into trypsin. Chymotrypsinogen will convert into chymotrypsin. Procarboxypeptidases will convert into carboxypeptidases. All three are responsible for digestion of protein. And then it also secretes some enzymes. So enzymes means active enzyme. They are not proenzyme. So active enzyme which are secreted by these cells of acne, or we can say this from the pancreatic part. These are the elastases. They digest elastin protein. Pancreatic amylases. Pancreatic amylases. They are responsible for digestion of amylase. DNAs. Digest DNA. RNAs. Digest RNA. Pancreatic lipase. Digest fat. Pancreatic lipase, digest fat. So these are the actual enzymes which are secreted by the cells of acne or these pancreatic part. Clear? Please note it down. Take the screenshot, then we'll go ahead. You have to tell me after noting it down. Noted? Done? Please tell me. Samara, note it down.
1 minute.
Please let me know when you note it down.
Done, sir.
Okay, great. Fine. So please note it down. In pancreas. Pancreas is a grayish pink gland. Pancreas is grayish pink gland which weight about 60 gram, full stop. It is about 2.5 centimeter wide. It is about 2.5 centimeter wide and 12 to 15 centimeter long and 12 to 15 centimeter long. Full stop. The exocrine part of pancreas the exocrine part of pancreas secrete an alkaline secrete an alkaline pancreatic juice with pH value 8.4 full stop about
Sir, with pH value
8.4 with pH value 8.4 full stop about 500 to 800 to 800 ml of pancreatic juice about 500 to 800 ml of pancreatic juice is secreted per day by cells of acne. This one by cells of acne, right? You have already noted down the proenzymes and the enzymes which is secreted by. So these collectively three proenzyme and these are the enzymes. So these collectively are called this one pancreatic juice, right? Okay. So this was all about the digestive glands, right? Now let's start with the process of digestion, right? So you have to put it. You have to put the heading: the process or physiology of digestion. The process or physiology of digestion, right? So we are going to note it down the digestion of four component mainly, so which are being digested during the process of digestion with the help of these enzymes. So these four component, out of these four component, the first one is, put the heading first one. So the main heading is physiology of digestion. Physiology of digestion. In physiology of digestion, the first subheading is that first one is digestion of carbohydrate, means sugar. Digestion of carbohydrate, right? So that is the first heading. Now look, so digestion of carbohydrate start from oral cavity, like from our mouth. We have already discussed that inside the oral cavity, so that is in oral cavity. So in oral cavity, we have discussed that there are certain glands, and we call these glands what? Salivary gland. And what they secrete? They secrete actually salivary amylase. So what salivary amylase do, right? In oral cavity, what exactly salivary amylase do? So it digests starch. So starch means bread. So we eat bread, grains and all that, right? Starch, the salivary amylase start working on it. Salivary amylase, right? So salivary amylase break down this starch into maltose and isomaltose. Maltose, isomaltose, and alpha dextrin plus alpha dextrin, right? So that kind of digestion takes place in our oral cavity, right? Salivary amylase, I want to mention one more thing. I have already discussed that salivary amylase is also known as xylem. I have already told you while telling you gland part, right? So this was the salivary amylase. So first, the Like here, I want to ask you a question. Like, which animal do not secrete saliva? Hey, come on, guys. I have explained you.
Sir, the ruminants, right? The cow.
Exactly. Very good. Ruminant, right?
Herbivorous animal. Herbivorous animal.
Yes, very good. Herbivores animal. We have discussed that. Please keep in the
Yes,
mind.
yes.
Yeah, great. So now the digestion takes place in the stomach, right? So in the stomach, hardly the digestion takes place, right? So mainly now digestion. So hardly there is any enzyme which digests any starch or any other kind of the carbohydrate part in our stomach. So now the mainly digestion second part, the digestion of the carbohydrate takes place mainly in small intestine. So in the small intestine, right? So what happened with the carbohydrate in the small intestine? Okay. So here the starch again. Here that is pancreatic amylase. So pancreas secrete pancreatic amylase. Pancreatic amylase breaks down it maltose again, maltose plus isomaltose plus alpha dextrin. Same thing which we have discussed ever, right? So in a small intestine, first starch is broken into broken to maltose, isomaltose, and alpha dextrin. Again, so next point, maltose is this maltose. This maltose is broken down into glucose with the help of enzyme maltase, right? Isomaltose, this one, second one, this isomaltose is broken down into glucose with the help of isomaltase. Isomaltase. Sucrose is broken down into next enzyme. Next. Sucrose is broken down into glucose and fructose with the help of enzyme sucrase. Lactose is broken down into lactose is broken down into glucose galactose. Glucose plus galactose with the help of enzyme lactase. Please note that. And wherever you feel the problem, let me know, please. So digestion of carbohydrate takes place at two places. First at mouth and second at small intestine.
Sir, what is the first one? Amylase, maltose, then maltose plus.
Maltose plus isomaltose and alpha dextrin.
Okay.
So what is written above salivary amylase?
Salivary amylase, ptyalin, that is ptyalin. Like I have mentioned that, when we were discussing salivary gland, we have mentioned that salivary amylase is also known as ptyalin. Both are the same thing.
Done, sir.
Done? Okay, great. Done? Should we move ahead? Okay. There is one more enzyme which is found in small intestine which work for the digestion. That is the next enzyme. That is, so the next one is, that is alpha dextrin. This one, look. Still alpha dextrin need to be broken down. This one, alpha dextrin, at both places, it need to be broken down. So again, alpha dextrin is broken down into glucose. Again, the enzyme is alpha dextrinase. So alpha dextrinase broken down into glucose. Now look, every component of the sugar have been converted into glucose, fructose, galactose, and all these are simple sugar. They can be absorbed by our small intestine, right? So these all sugar have been converted into simpler form, which can be absorbed and utilized by our body. Clear?
Yes, sir.
So the next heading is digestion of protein. So second part is digestion of protein. Our oral cavity do not have any role in the digestion of protein, right? So the digestion of protein start from stomach only, right? So first, at the stomach. So what are the enzymes and what are the component in which it has been broken down? So the first, there is a proenzyme called pepsinogen. We know the pepsinogen is secreted by digestive glands, right? That means gastric glands, right? Pepsinogen is a type of proenzyme, means it is inactive. Whenever the HCl is secreted by our digestive glands, the pepsinogen get converted into active form pepsin, right? So that is the active form. I want to tell you the cola drinks, they are actually, these are the cola drinks, they help in digestive process, right? Actually all these black colored cola drinks, they have pepsin enzyme, so it help in digestion. That's why normally people take these cold drinks during the their meal process, right? So because there are two factors, these cold drinks have dissolved carbon dioxide, so this carbon dioxide breaks down food, it help in breaking down food. And the second, pepsin enzyme that help in the digestion of protein part. Okay, so next, the proteins are now broken down into peptides with the help of this active pepsin. So pepsin break down protein into peptides. Again, there is so this was one process. Again, there is another enzyme that is called prorenin. This is also in active form. Prorenin. So prorenin is again inactive. In presence of HCl, prorenin is converted into renin. Now, what exactly renin do? The milk protein that is called casein. So casein, renin break down casein enzyme. So I have told you while I was telling you the gastric glands, I told you that the renin is the enzyme which is secreted in the young mammals, but like adults like us, it is not secreted in our body. When we grown up, when we become adult, so it is stop getting secreted, right? So they break down casein. That casein is a milk protein which gives white color to the milk. So casein is milk protein. Renin break down casein into paracasein. So it break down casein into paracasein. Again, paracasein this paracasein in presence of pepsin, it is broken down into peptides, right? Note it down. So that is the digestion of protein which takes place in our stomach.
Done, sir.
Great. So next,
Done.
done or
Yes, sir, done, sir. Done, sir.
okay. At the small intestine, right? So how does the digestion of protein takes place and is the small intestine? Now the trypsinogen, so they have again a proenzyme. We have discussed few minutes before. Trypsinogen, that is an inactive enzyme. So trypsinogen is converted into trypsin. Which part convert trypsinogen into trypsin? So there is an enzyme that is called enterokinase. Enterokinase convert trypsinogen into trypsin, means inactive form into active form. Right? Look. Now this chymotrypsinogen is converted into chymotrypsin. With the help of trypsin. Proteins are broken down into dipeptides with the help of trypsin. Elastin protein, it converted into dipeptidases by a protein called elastases. Sorry, enzyme called elastases, right? Now again, the dipeptides, they are broken down into amino acid final component. They are broken down into amino acid by amino peptidases. Sorry, just a minute. Not dipeptidases. Aminopeptidases. Dipeptidases are broken down into amino acid with the help of enzyme dipeptidases. Dipeptidases. And peptides are broken down into amino acid with the help of aminopeptidases. That is the digestion of protein which takes place in our small intestine.
Done, sir.
Done. Now the next part is put the heading digestion of fat. Digestion of fat. Look, the fat is bile juice, right? What actually bile juice did, so it emulsify fat. Are you aware with the process of emulsification?
Yes, sir.
Okay. So
Happens in the-
emulsify.
Yeah, by
Yes.
juice.
Right, missiles. It mix it into water, right? So that it causes the emulsification of fat. Now the emulsified fat is emulsified fat. It is converted into fatty acid and glycerides in presence of lipase, right? So lipase, it breaks down into fatty acid plus diglyceride. Right? Again, diglyceride is broken down into fatty acid plus monoglyceride. Again, the function of lipase does it. Monoglycerides is further broken down into fatty acid and glycerol. Fatty acid plus glycerol with the help of lipase. So digestion of fat takes place only in our small intestine, right? It does not take place at our stomach. So that's why the fatty things get digested at uh, in longer duration, right? It takes longer duration. Note it down. Then we will do the last part, that the digestion of nucleic acid.
Done, sir.
Okay, great.
1 minute, sir.
Yeah, sure.
Done.
Digestion of nucleic acid. Nucleic acid, that is DNA and RNA. So whatever we eat cell. So cells always have DNA and RNA. They always have nucleic acid. So how the nucleic acid is being digested? So what actually the digestion of like DNA and RNA, that nucleic acid, first it takes place by pancreatic juice. Pancreatic juice. Like few minutes before we discussed that pancreatic juice secrete these enzymes. So pancreatic juice, what it do? It breaks down DNA into nucleotides. With the help of enzyme DNase. It breaks down RNA into nucleotides with the help of enzyme RNase. Right? Again, it is also digested by the intestinal juice, but digestion takes place only in intestine part. So, intestinal juice. How does intestinal juice does? So, what it do? It breaks down again nucleotide into nucleoside and phosphate. So with the help of enzyme nucleosidase, with the help of enzyme nucleosidase. Again, nucleosides, this part. Nucleoside is broken down into nitrogen base plus sugar, pentose sugar. That is a simpler form with the help of nucleosidase. Right? So that is all about the physiology of digestion, right? Please note it down.
Done, sir.
Okay,
1 minute, sir.
fine. So now the next part is absorption.
1 minute.
Okay, please note it down. Note it.
Done, sir.
Okay, so please put the heading absorption of nutrient. I will show you two diagrams and this will complete the story, right? And remember these two diagrams. The first diagram, it is going to help you a lot in your medical examination. So please take the screenshot and like keep it in your mind. This is like my handwritten diagram. Look, this is schematic diagram. So I have made a diagram of this complete digestive tract. Look, stomach, duodenum, jejunum, ileum, and long. In a stomach, absorption of water, alcohol, some monosaccharide like glucose, some drugs and salt takes place. Alcohol is the fastest absorbing substance by human intestinal gut, or we can say human gut, right? So alcohol is the fastest thing which can absorb by our body. And even whenever we drained off and feel a lack of energy, what we do, you just take a 2 spoon or 3 spoon of glucose, you dissolve in it water and take, and you feel energetic. Why? Because glucose is the simplest form and stomach is able to absorb this glucose component, then that's why you feel the energy whenever you take the glucose, right? Drugs are also absorbed by stomach. Duodenum, it absorbs iron, calcium, and amino acid. Jejunum, it absorbs monosaccharide, amino acid, fatty acid, vitamins, and water. Ileum, it absorbs vitamin B12, bile salt, and water. Large intestine, water, some minerals, largely NaCl, drugs, vitamin produced by the bacteria are absorbed by the large intestine. Clear?
Yes, sir.
Take the screenshot. Done. Atif,
Yes, sir. Done.
are you getting everything? Okay. So one more diagram. Yes. Somebody is asking something. I'm able to listen. Who is speaking? Is there someone asking me anything?
No, sir.
Okay. Fine. One more diagram. You will get each and everything about the absorption in this diagram. This is also my handmade diagram. I have tried to show complete process, complete picture over. Look, this is the complete picture of absorption. Look. So first, polysaccharide. Polysaccharide means complex sugar. It is broken down into glucose in process of digestion. Again, disaccharides, lactose, sucrose, and maltose, and fructose. Look, glucose is absorbed over glucose and galactose, absorbed by these are the villi actually. This is the structure of villi. Let me explain you first the structure of villi. Look, this is the structure of villi. And let me tell you, the upper part is made up of this upper part. This upper part is made up of epithelium, right? Please try to understand. This upper part is made up of epithelium, right? So that's why I have tried to show in green color. So this green color part, this part is made up of epithelium. Inside this part, there are blood capillaries. Look, these are the blood vessels, right? So these are the blood vessels which are found inside. Look, which are found inside this one. These are found inside the villi part, right? Inside the blood capillary, there is lacteal. What is lacteal actually? This part. I want to show you. I will show you in blue color. This part. This part is actually lacteal, right? I have shown in blue color. This lacteal. What is lacteal? Lacteal is actually lymph vessel. This is lymph vessel. So look, fat is actually not absorbed by these blood capillaries because it is made up of a large droplet, right? So fat is absorbed by lymph vessel, rest amino acids and DNA, RNA, amino acids, sugar, and rest of the things are absorbed by blood vessels. Look, now the first come to the polysaccharide or glucose. So glucose and galactose are absorbed by active transport. Active transport means spent of energy takes place, expansion of energy. So ATP will be utilized over here and it is taken by villi, right? So fructose is taken by facilitated diffusion. Facilitated diffusion means this is the passive absorption. No expense of energy takes place. So that's how the absorption of sugar taken place by the surface of villi. Again, second thing is fatty acid or fat. So fat is converted to fatty acid and glycerol. Fatty acid and glycerol both are absorbed by small these villi with the help of simple diffusion process. Now come to the protein. Protein is broken down into peptides, then amino acid. Amino acid are absorbed by active transport. Mean here also the ATP is required for the absorption of these amino acids. Fourth, water is absorbed by osmosis, means no energy is required over. Most water soluble vitamins are absorbed by simple diffusion, right? And over the fat is actually absorbed by, look, let me tell you, fat is actually absorbed by these lacteal, right? That's how the absorption of mineral nutrient takes place in the villi of a small intestine. Is it clear or not?
Yes.
If it's clear, please take the screenshot, and I like expect that after the class you people will draw those diagrams which I have drawn with my hand. So these are very important, and merely with the help of this diagram you can this diagram and previous diagram you can solve each and every question which is asked in medical examination, and you will get ample of the questions from this chapter and number of the question from these diagrams. You will be able to solve all questions from these diagrams, right? So note it down some point. Glucose and galactose are absorbed by active transport. Glucose and galactose are absorbed by active transport. Full stop. Fructose is absorbed by facilitated transport. Fructose is absorbed by facilitated transport. Full stop. Next point. The most rapidly transported monosaccharide is galactose with glucose.
So the most
rapidly transported monosaccharide is galactose with glucose at second position. Next point. Amino acids are absorbed by.
Can you repeat?
Amino acids are absorbed by active transport mainly, but some of the amino acids are also absorbed by the passive process. Next, about 90% of water, about 90% of water absorption occur at small intestine. About 90% of water absorption occur at small intestine by the process of osmosis, full stop. Absorption of water also occur at stomach and large intestine. Next, note it down. The fat is converted into micelles. What is micelles? Come on, you must have studied it in class 10. Nothing, yeah. Micelles means water, that is a mixture of water and fat, that is called micelles, right? So mixture of water and fat is called micelles. Suppose that you have like fat, like oil and water in a bottle. You shake it, but it will always remain separated. So that is a water, like fat will be at upper part and water will be at lower part. Put a half spoon of surf excel or any washing powder and shake it. Now the fat will be completely mixed into the water. That dirty kind of the liquid will be called micelles, right? So fat is converted into micelles with the help of bile juice or bile salt. Again, what happened? Micelles are converted into a small droplets, that is called chylomicrons. Full stop. Micelles is converted into micelles is converted into a small droplet by ER called chylomicron.
Sir, what by ER?
ER, endoplasmic reticulum, by ER called chylomicron. Full stop. These chylomicrons are absorbed by lacteal. These chylomicrons are absorbed by lacteal with the active process. Next,
So this chylomicrons are
absorbed by lacteal. Right? Next, absorption of vitamins vitamins and electrolyte also takes place by passive process. But some of them are also absorbed by active process. Last but not the least, that is the last process. Taken this screenshot. Should I remove this slide? I'm going to remove it, right? Look. The last process is egestion. Egestion must not be confused with excretion. The removal of waste food is called egestion. Please note it down. The elimination of feces. The elimination of feces from alimentary canal is called egestion or defecation. Full stop. So this was all about this one. And last thing is that some disorders. So there are some disorders like digestive disorders. So put the heading digestive disorders. So disorders of digestion. Disorders. So the first disorder is jaundice. Jaundice, you know, this eyes, white color of eyes ball, it become yellow, and like our nails become yellow. Liver get affected, and it is spread by the dirty water. Right? Note it down. Jaundice is a yellowish coloration of whites of eye. Jaundice is yellowish coloration of whites of eyes, comma, skin and mucous membrane due to buildup of yellow color component bilirubin. We have studied that bilirubin is a component of bile juice. It is an excretory substance. We have already discussed. So bilirubin must be excreted out from our body. So whenever this bilirubin start accumulating in our blood due to dysfunction of liver, this is called jaundice, right? So next point. Jaundice is caused by infection in liver, from dirty water, Next disease is vomiting. Is it's like note it down. Second name B is vomiting. Note the skeletal muscles of abdominal wall and diaphragm the skeletal muscle of abdominal wall and diaphragm contract, increases intra-abdominal pressure due to which
Increases extra.
extra pressure to increases intra-abdominal pressure, increases intra-abdominal pressure, due to which sphincter relaxes, full stop. As a result, content forced to upward through esophagus. Next, C. Diarrhea. Is also caused by bacterial infection. Note it down. Diarrhea is increase in the frequency of vomiting, increase in frequency, volume of fluid content, increase in frequency of volume and fluid content.
So can you repeat, sir?
Diarrhea is increase in frequency, comma, volume and fluid content of feces, caused by increased motility, and decreased absorption by the small intestine.
Sir, increased
Motility and decreased absorption by small intestine. Full stop. Frequent diarrhea may lead to dehydration. Next, constipation. It is infrequent or difficult defecation caused by decreased motility of the small intestine. Right? Next, indigestion.
small intestine indigestion.
Next is indigestion. Indigestion. Incomplete digestion is usually accompanied by one or more of following symptom, that is pain, nausea, vomiting, acid regulation, gas, etc. So that's it for this chapter, digestion and absorption. Is it clear to all of you? If you have any
Yes,
doubt
sir.
in this chapter, please let me know.
Yes.
I'll explain it for you. Any one of you. I want to reconfirm from people. Atif, did you got all things, all points, whatever we have discussed in this chapter?
Yes.
Okay, great. Minashi,
Yes.
Azia, clear? Right. Okay. So we are going to start with the next chapter, that is the breathing and exchange of gases. Please put the heading breathing and exchange of gases. So give me one second. Breathing and exchange of gases. Yes. So let me explain you the first and foremost, that is the human respiratory organ. Respiration. Yes. Let me show you diagram. So put the heading human respiratory system. Human respiratory system. Right. One second. Look, this is human respiratory system. It's very basic kind of diagram. You all know about this. Look, this is nasal cavity, and these structure are actually called concha, nasal concha, and this one is nostril, from where we take like these are the holes are called actually nostrils. The space inside the nose is called nasal cavity, and this space inside our mouth, this is called oral cavity. As we take oxygen from both of the organs, suppose that if I close down my nose, then I will start breathing from my mouth. That is not an effective way of breathing. So generally the effective way is from nasal cavity. The part till both of the part, like our oral cavity and our nasal cavity, can remains connected, that is called pharynx. After that, what happened? The part which is responsible for breathing, that is called trachea. And above the trachea, there is an organ that is called larynx. Larynx is actually called sound box. With the help of larynx, we are able to speak, right? If larynx is not available over here, then we won't be able to speak. Below the larynx, there is a trachea. Trachea is like that is called windpipe or respiratory pipe. Trachea is made up of cartilage, right? This is not a complete cartilage ring. This is this type of the cartilage ring. This is actually incomplete at the back side. So this is not complete cartilage ring, but trachea is the cartilage. Trachea is made up of cartilaginous rings. They protect trachea being collapsing. Again, trachea divided into two parts, that is called bronchus, right bronchus and left bronchus. Bronchus is singular term, bronchi means plural term. This is right lung and this is left lung. Left lung is a bit smaller than right one, right? There is a curtain-like structure that is called diaphragm. Diaphragm is that curtain-like structure which separate this digestive cavity from the this one, respiratory cavity, right? So take the screenshot, then I will show you one more diagram. Please take the screenshot. Now let me show you another diagram. Now these are the actually human lungs, right? So this is diagram of human lungs. Look, the left part of the brain, the left part of the lungs is smaller than right. The reason behind it that this part is covered by heart. So this part is covered by heart. This part. So what we call this part where our heart remains situated. So a space is there for our heart. This part. So this part is called cardiac notch. Cardiac notch. So heart remains fit at this point. Cardiac notch. Cardiac notch, right? And the space between the two lungs is called mediastinum, right? The space between two lungs. So let me, the space between two lungs, this space that is called mediastinum. This complete space. Mediastinum, right? So this is called mediastinum. Again, so right heart have these three lobes, right? And the left heart have these two lobes, right? Again, I'm sorry, the right lungs have three lobes and the left part of the lungs have two lobes, right? The lungs remains covered with a special kind of the membrane. That membrane is called pleura. So pleura is a type of membrane and it is made up of epithelium, right? Inside the pleura membrane, there is a fluid called pleural fluid. Lungs, they expand and they like contract inside the pleural membrane. It protects the lungs, delicate lungs from friction. right? They protect it from being ruptured. Again, the trachea is divided into two parts, that is left and right bronchus. Bronchus is further divided into bronchi, and bronchi end up with these balloon-like structure. These balloon-like structures are called alveoli. Alveoli are actually the part where the exchange of gases takes place. The exchange of like oxygen is taken over here, and carbon dioxide is given by the blood capillaries at this place. Clear?
Yes.
All clear? Okay.
Yes,
So
sir.
please put the heading human respiratory system. Right lung is larger and of 700 gram, and left one is smaller and of and of 600 gram. Full stop. Keep on writing in bullet form. Next point. There are 300 million alveoli. There are 300 million alveoli found in lungs. Full stop. Keep on writing in this same point. Alveoli are balloon-like structure. Alveoli are balloon-like structure where exchange of gases. Alveoli are balloon-like structure where exchange of gases takes place. Next point. Lungs are enclosed in two membrane. Lungs are enclosed in two membrane. Outer parietal pleura, outer parietal pleura, and inner.
Sir, lungs are enclosed in two membrane.
Two membrane, outer parietal pleura and inner visceral pleura. Inner visceral pleura, which closely invest the lung. Full stop. In the same point, pleural fluid is filled inside this space, and it is secreted by pleura membrane. Next, vocal cord, right? Vocal cord means the sound box, right? So vocal cord. So what is the vocal cord? Actually, inside the larynx, there are two membranes. One is longer one, so not two membrane, four membrane. Actually, two pair of membranes are found. One longer one, another pair is shorter one, right? Whenever the air passes through it, they start vibrating, and because of this vibration, we are able to speak, right? So these vocal cords are actually the part of larynx which help in talking or which help in producing sound, right? Next point, vocal cord. Vocal cord. Inside the larynx, there are present the vocal cord. Full stop. These are two pairs of fold of. These are two pairs of fold of mucous membrane that extend into
Sir, these
these are two pair of fold of mucous membrane that extend into lumen of larynx. Full stop. The upper pair is termed as false vocal cord, and lower pair is called true vocal cord. Next point.
sir, upper pair is called
Upper pair is called false vocal cord, and lower pair is called true vocal cord. Underline the term false vocal cord and true vocal cord. These are the questions. Next paragraph. The vocal cords are usually, it's very, very important point. Please make a study this question for your medical examination. The vocal cords are usually thicker and longer in men than women. Vocal cords are usually thicker and longer in men than women, and therefore they vibrate slowly. Full stop. Thus men have lower pitch than female. One last very important point in vocal cord, put a star mark, is also a question for your medical. Vocal cords are made up of yellow elastic tissue. Full stop. Whenever air passes through them, sound is produced. Third heading is trachea. Third subheading is trachea. As I have told you, they are made up of cartilage rings, and cartilage are not complete, they are incomplete. So they are incomplete rings, they are not connected at the back side, so they are incomplete kind of the ring. So these kind of the rings, they make the trachea, right? Trachea bear or ring, underline. It has been asked in medical examination, which cartilage makes trachea? In animal tissue, we have studied. Trachea bear rings of hyaline cartilage, which are incomplete posteriorly. full stop. They prevent trachea. They prevent trachea during inspiration. They prevent trachea during inspiration. The inner part of trachea is made up of ciliary epithelium and mucous cell. Ciliary epithelium and mucous cell. What happens, because whenever the dust particle, whenever the any carbon particle, when we inhale it. So what happens? These mucus trap them, and these ciliary they start motion like them. And then what happens? We feel cough, and because of coughing, we spit it outside. So that is the or else if more dust and more fumes is there, we start sneezing. So by the process of sneezing, these cilia help it to like throw it out of our body. So that is a defense mechanism which is found. One more important function which I want to tell you, that these ciliary epithelium do not have capacity of regeneration. So what happened? Whenever a smoker starts smoking, they start dying, and in a smoker they die completely, but they never regenerate, right? So smokers are more prone for respiratory diseases, right? Because of the death of the ciliary epithelium. Is it clear to all of you? Did you got it? Okay. Next, put a star mark, and there is a two more point. What is the normal human breathing rate in humans? Can you tell me what is the breathing rate in human? How many times we breathe in a minute?
Sir, 15 to 18.
Yeah, almost 16, 20. Note it down. At rest, the breathing occur around. At rest, breathing occur around 14 to 18 times per minute. At rest, breathing occur around 14 to 18 times per minute. In normal man, so this was something about the respiratory system of a human being. Is it clear to all of you, or do you have any doubt? Clear?
Done, sir.
Okay, great. Now there is a topic called respiratory volume, respiratory volumes. Then we will study the respiratory mechanism of respiration. So respiratory volumes. So put the next heading, that is the respiratory volume. Respiratory volumes. So, first one is tidal volume. Look, what is tidal volume? Tidal volume is also called TV. Right? So the volume of air that we inspire or expire normally is called TV, tidal volume. So this is the 500 ml. Like normally we are talking with each other, we are studying, like I'm teaching you, you are like studying. So the gas or the gas, not the oxygen, actually this is the gas or air we inhale and exhale, that is approximately 500 ml. So that is called TV, tidal volume. Note it down. The volume of air inspired or expired. The volume of air inspired or expired, during normal breath, full stop. This is about 500 ml. This is about 500 ml. Full stop. A healthy man can inspire or expire. A healthy man can inspire or expire 6,000 ml. A healthy man can inspire or expire 6,000 ml to 18,000 ml of air in a minute. Fine. So before going further, I have a question for you. I have a question with you. Actually, I want to discuss that thing with you. So when you have your your final examination, approximately date, can you tell me? Amira, Istisham. Come to examination. Yes, Istisham. Amira.
Sir, end of February.
End of Feb, right? And Mazia, Alvia, Atif, all of you can tell. Feb, okay. So I think at the end of Feb. And I think the syllabus will be plant physiology as well as human physiology, right? Both of the physiologies will be the syllabus.
Yes, sir.
Great. We'll do that. Actually, I want to make an idea in my mind. So I'll take the classes accordingly. Next. So this is the TV clear, tidal volume clear.
Yes, sir.
Now the okay. Next is residual capacity, residual volume. Residual volume. Residual volume is RV. So what is residual volume? So the volume of air which we cannot inspire, we cannot expire out. If even we forcefully expire, still some amount of the, if I put complete force on my lungs and I try to push out the complete air out of the lungs, still this some amount of air is not going out of the lungs. It will always remain ill, else the lungs will stick and I'll collapse. So that amount of the air which remains inside the lungs all time, it cannot be taken out from the lungs naturally. So that part of the air is called tidal volume, TV. Residual volume, RV. Right? Please note it down. The volume of air which remains, the volume of air which remains is still in lungs, is still in the volume of air which remains is still in lungs after a forceful expiration. Full stop. This is about 1100 ml to 1200 ml. So every time minimum there will be 1100 to 1200 air inside our lungs. We cannot pull it out this air out of our lungs, right? So this is called RV, residual volume. Next, third is inspiratory capacity. Inspiratory capacity. Inspiratory capacity is called IC. Please note it down. This is the look, before inspiratory capacity, I want to tell you two terms. Actually, it is not mentioned in your NCRT. So, because inspiratory capacity have a formula. So before going into inspiratory capacity, I want to tell you what is IRV, inspiratory reserve volume. So put the heading. Then I will tell you inspiratory capacity. Inspiratory reserve volume. So inspiratory reserve volume, this is called IRV. Note it down. The extra volume of air that can be extra volume of air. Just give me 1 second. 1 second.
The extra volume of
Extra volume of air that can be inspired forcibly after a normal inspiration is IRV. It is 2500. It is 2500 to 3000 ml. Look, what is IRV? So I have told you that tidal volume is what? Like I am breathing in, breathing out. That is the 500 ml in one go, right? So 500 ml. So that is TB is 500 ml. That is I am taking in 500 ml normally. After the 500 level, after this 500 ml, if I take deep breath, like I take extra forcefully this way. So the extra volume which I take in, that is 2500 to 3000 ml. So that is called what? IR. Clear?
yes, sir.
Okay. Next, ERV. Expiratory reserve volume. Expiratory reserve volume. That is the opposite of ERV. IRV. That is ERV. Expiratory reserve volume. Right. So it is extra amount of air which I can expire after normal expiration. That is called ERV. IRV, I can inspire after normal inspiration. That is IRV. ERV, I can expire after normal expiration. That is called ERV. So please note it down. It is the extra amount of air that can be forcefully forcefully expired after, that can be forcefully expired after normal expiration, after normal expiration, right? Now put a fifth heading, and that is,
So there's no quantity for this?
right?
No quantity for this?
Yes, yes, there is quantity. It is about 1100 ml to 1200 ml, right? So look, now come to the inspiratory capacity, IC. Inspiratory capacity, right? That is IC. So what is the IC? IC is the total volume of air a person can inspire after a normal expiration. Please note it down. Total amount of air a person can, total amount of air a person can inspire after a normal expiration. So how much a person can inspire after normal expiration? First, PV, tidal volume, then look, first PV 500 ml, then extra, that is IRV. So formula is IC is equal to TV plus IRV. How much? IC, that is 500 ml, and plus IRV, how much? This is 2500 to 3000. So what can be IC? What will be the range for the IC? So IC will be from 3000 ml to 3500 ml. Is it correct or not? Clear?
Yes.
Right? So please note it down first. Then we'll go in next. Note it? Okay. Look. Next is the sixth one is, that is expiratory capacity. Expiratory capacity. How much a person can expire after normal inspiration, right? There is EC. So please note it down. The total volume of air a person can expire, total volume of air a person can expire after normal inspiration. So what is the volume which normally I take, that is TV, plus the forcefully which I can expire, ERV, right? So TV is 500 ml, and we have studied ERV is 1100 to 1200. So how much EC? It will be 1600 to 1700 ml. Is it correct? Clear? Note it down.
Yes, sir.
Okay, great. So the next, seventh point. Seventh one is functional residual capacity. Functional means that is workable, which function. Functional residual capacity, which remains in our lungs. So this is called FRC. First note it down, then I'll explain it. The volume of air, so this is 5 after leaving 500, whatever remains, that is FRC. The volume of air remain in lungs after the volume of air remains in our lungs after normal expiration is called FRC. So how much it will remain? I have expired. How much I have expired? Equal to tidal volume. That is 500 ml. What I expire normally. So suppose that I have expired 500 ml. So how much air inside? So that is equal to residual volume 1100 to 1200, which cannot be taken out of the lungs. Then extra, which I can forcefully expire, that is ERV. Clear or not? So how much residual volume? I have told you RV is 1100 ml to 1200 ml. What is ERV? Again, ERV is also 1000 to 1100 ml, right? So we can say this is 2100 ml to 2300 ml. Correct?
Yes.
Note down, please.
Yes, sir.
What is vital capacity? Put it on vital capacity, VC. You will get ample of the questions in your medical examination from these 9 terms, and no question will be out of these 9 terms, I claim. So you have to like revise it again and again, like 6 to 7 times. Then you will be able to assimilate it in your brain, right? Vital capacity. The maximum volume of air, note it down. The maximum volume of air a person can breathe out, maximum volume of air a person can breathe out after a forceful expiration or maximum volume of air or maximum volume of air a person can breathe out after forced inspiration.
Inspiration or expiration?
Inspiration. Is called vital capacity. So vital capacity is equal to total lungs capacity and minus the volume which cannot be breathed in, breathe out, residual volume, right? So this is the total, this is the VC. Ninth and last one is TLC, total lungs capacity, that is TLC. So TLC is equal to vital capacity is complete plus residual volume, right? This is the actually formula. Note it down. The total volume of air present in lungs and respiratory passes.
The respiratory
Passes. This trachea, bronchi, bronchioles is
Yes.
called TLC. Please take the formula. And this
Yes.
is all about the respiratory volumes. So we have completed today till respiratory volumes and put the heading in for the next class. Next class we are going to do exchange of gases.